Hi, in today's session, we are going to talk about the organic compounds and carbohydrates. What are organic compounds? All living organisms are made of biological molecules, which determine how they grow, function, and reproduce. So, these molecules are mainly organic compounds, meaning they contain carbon. But organic compounds also have hydrogen, oxygen, sometimes nitrogen, sulfur, and phosphorus also. Apart from water, nearly all the material in your body is organic in nature. So why do we consider carbon as the element of life? Carbon has a 4-valence electron, so let it form 4 covalent bonds with other atoms. So these bonds are stable and very strong. So this structure made carbon ideal for complex molecules. So the carbon atoms can bond with other carbon atoms to form straight chains like fatty acids, branch chain, and rings like glucose. So this flexibility creates an almost infinite variety of molecules. 3D shapes and carbon's versatility. So each carbon atoms form a tetrahedral arrangement of bonds, like a pyramid with a carbon at the center, as you see in this picture. So this allows for spatial diversity. So the molecule can twist and fold into various 3D shapes. So some are linear, others are branch, and some form closed ring or even complex helices. So this 3D shape affects how molecules interact with enzymes and receptors. Monomers and polymers, building blocks of life. Monomers are small simple molecules like glucose, amino acid, nucleotides. And many monomers join together through chemical bonds to form polymers. So monomers join with each other and form polymers. So polymerization means the process of linking monomers into long chains. So example for polymers are carbohydrates. So glucose join together with each other and form carbohydrates. And the amino acids go through with each other and form proteins. And the nucleotides combine with each other and form nucleoacids. Carbohydrates, fuel and structure. So the carbohydrates are organic molecules. They are made up of carbon, hydrogen and oxygen. So their general formula is roughly CH2ON, where the N can vary. So the main functions of carbohydrate are they act as immediate energy source. So glucose can convert to ATP and it acts as energy storage like starch in plants, glycogen in animals, and carbohydrates have a structural role. So cellulose in plant cell walls, chitin in fungi, and carbohydrates form a huge variety of sugars, starches, and fibers. So we can classify carbohydrates into main 3 types. It is classified by number of sugar units. They are monosaccharide, disaccharide and polysaccharide. When they have a single sugar molecule, it's a monosaccharide. Examples are glucose, fructose, galactose. When there are 2 monosaccharides combined with each other, we call it as disaccharide. So examples are sucrose, lactose. When there is a long chain of sugars, we call it as a polysaccharide. The examples are starch, glycogen and cellulose. Monosaccharides, they are the simple sugars. So they are the simplest carbohydrates, like single ring molecules. The ratio is carbon to hydrogen to oxygen is 1 to 2 to 1. So the general formula is same as previous. So we can classify the carbon monosaccharides according to their carbon number. When there is a 3 carbon, it calls as a triose. Example is glyceraldehyde. When there are 5 carbons, we call it as a pentose. Example is ribose, deoxyribose, like we can see them in DNA and RNA. When there are 6 carbon units, we call it as hexose. Examples are glucose, fructose, galactose. You can see here, when there is 3 carbon unit, it is a triose. When there is 5 carbon units, it's a pentose. When there is 6 carbon unit, it's a hexose. The structure of hexose sugars. Hexose often form ring-shaped structures in solution, like in this picture. So the numbering of carbons, 1 to 6 we can number the carbons. So it helps us to identify where atoms has attached. So when there is small difference in structure, it can lead to isomerism. So there is same formula but different arrangement. So examples are alpha glucose versus beta glucose. They differ only in orientation of the hydroxyl group on carbon 1. So these tiny changes affect how polysaccharides are formed. So there is alpha glucose and beta glucose. The function is also different. In alpha glucose, they use for energy storage like starch and glycogen. But beta glucose is used for mainly for structural uses. They form mainly cellulose. So therefore, it's a small structural change, but there is a huge biological difference. So this demonstrates the structure and function relationship fundamental to biochemistry. So you can see here alpha and beta glucose, disaccharides. When the two sugar units combine with each other, the disaccharides are formed. So this reaction join through a condensation reaction. So a molecule of water is released during bond formation. A bond formed is a glycosidic bond. It's a type of covalent bond. And the examples are when glucose combined with fructose, it forms sucrose. When glucose combined with galactose, it forms lactose. When glucose combined with another glucose, it forms maltose. You can see the diagrammatic illustration here. Glycosidic bonds in detail. So the glycosidic bonds are named by which carbon atoms are linked. So when there is a 1,4 glycosidic bond, it's usually a linear chain. And when there is a 1,6, it's a branching point. So the type of linkage affects the structure and digestibility. So structure is straight or branch. The common disaccharides. The sucrose, it's as we talked previously, it is combined when the glucose and fructose combine with each other, sucrose is formed. So it is a non-reducing sugar. And the lactose, it is important in infant nutrition. And the maltose is also a reducing sugar. To identify reducing sugar, we use Benedict test. So the procedure is first we add Benedict solution. It's usually blue in color to sugar sample. And we heat it in a water bath for about 3 to 5 minutes. Then we observe the color change. When there is this reducing sugar present, we can observe blue to green to yellow, orange, brick red color change. The result is brick red. So when there is a reducing sugars are present in the sample, it convert to brick red color. You can see the color differences here. Blue, green, yellow, orange, brick red. Brick red is the last result of this test. Testing for non-reducing sugar. And the sucrose does not react directly with Benedict solution because it's a non-reducing sugar. To test, we have to boil sample with dilute hydrogen chloride. So this hydrolyzes the glycosidic bond. Then we cool it and then neutralize the sodium bicarbonate. Then we repeat the Benedict test. So a positive orange to red color result confirms presence of monosaccharide after hydrolysis. So that's all the basics of organic compounds and carbohydrates. Thank you.